As we all know, CAR T-cells have revolutionized the treatment of CD19-positive hematological malignancies. However, many barriers still remain to their widespread applicability. These include not only high cost of autologous products, but also rapidly progressing disease, which may result in patients ultimately not being able to receive treatment due to deteriorating conditions, as well as poor T-cell fitness in the starting material of heavily pretreated patients. Therefore, off-the-shelf allogeneic CAR T-cell products, where the starting material for CAR T-cell manufacture is taken from an healthy donor, are highly desirable. But their development still requires overcoming limitations which are associated with graft-versus-host disease due to the TCR expression on the CAR T-cells from the donor, as well as the poor persistence of CAR T-cells in allogeneic setting. To overcome this barrier, at UCL, Martin Pule and Paul Machacha have developed a simple protein-based approach for generation of TCR-negative CAR T-cell products without the need for genome editing. This approach is based on the KDEL anti-CD3 module, which it's an antibody conjugated to tagged to a KDEL motif, which binds to the CD3 and retains it in the Golgi compartment inside the cells, thus preventing the TCR-CD3 complex to assemble on the cell surface. This approach has several advantages, as both the CAR and the KDEL module are expressed in one construct, so no genome editing is required. Therefore, there is no need for complex and expensive genome analysis for its use in the clinic. In the KCAT19 trial, we use core blood-derived T-cells as a starting material, with the idea of using T-cells which present with a better phenotype, which has been associated with better CAR T-cell persistence and clinical efficacy. Cells are modified to express an anti-CD19 CAR, which is based on the KCAT19 binder, which was originally developed here at UCL and has been recently submitted for FDA approval by Autologous Therapeutics, together with the CD3 KDEL module. And with the source, and we've also included a source suicide gene, which was incorporated as a safety switch. In our group, we have developed a semi-automated closed system for production of KCAT19 cells, which uses the Clinimax prodigy for selection, activation, transduction, and expansion of core-derived blood-derived CAR T cells. We have additionally established a strict method to obtain CD3 and TCR-negative cells. And we have developed this system to deplete any residual CD3 TCR-positive cells using the Clinimax Plus. We have shown that KCAT19 products from core blood-derived T cells expand really well and are constituted mainly by naive memory T cells, which have been shown to have improved clinical responses, and these cells are effective against CD19-positive targets. UCL is now sponsoring a clinical trial called KCAT19 to evaluate safety and feasibility of this allogeneic product in patients with CD19-positive malignancies which have failed prior treatments and are either not eligible or have failed previous autologous CAR T cells. And with this, we hope that this will constitute the first stepping stone towards the development of off-the-shelf allogeneic CAR T cell products.